Hi everybody, I'm Chloé. I am a postdoctoral fellow in the laboratory of Chloé Berger at Université Laval in Quebec City. And today I will talk about the ecology of eDNA, RNA, and how we can use it to track invasive species. So I was hired about 1 year ago to work on the iTrack DNA project. So if you were at the workshop today, Karen spoke about the iTrack DNA project. So this is a very big project that is funded by Genome Canada, Genome BC and Genome Quebec. And one of the main goals of iTrack is to develop innovative and accessible tools to study environmental DNA. It involves a lot of actors, for example, from universities, from the Canadian government, and also from native communities. And I am specifically working on the fourth objective, which is to experimentally study the ecology of eDNA and RNA. When we began the project, we had two main challenges, which were to understand how the biology of a species and its interaction with the environment could affect the probability of detection of these molecules. And also we wanted to understand which factors could influence the origin, the fate and the state of eDNA and RNA. As a matter of fact, there is currently a lot of variation in the quality of the data that is reported, and we understand very little about the factors that will influence the origin, the fate and the state of eDNA and RNA. And these gaps could impair data interpretation and the objectives in species monitoring, which is all the more important for invasive species. We decided to work on the goldfish. So as you may know, this is a real invasive species in many lakes and rivers in Canada. What happens is that when it invades new environments, it can disturb our fish communities, like it will eat their food, it will transmit diseases. And once it has invaded a new environment, it's very hard to remove it. So our objectives were to perform a series of experiments in controlled laboratory conditions in order to document the independent and the interactive influence of key biotic and abiotic factors on the origin, on the fate, and on the state of the molecules. So when I'm talking about the origin, we were interested in following the production rate depending on two abiotic factors that were temperature and pH. So we did that at 6 time points when the fish were present. For the fate, we were interested in following the degradation rate depending on the same abiotic factors, this time at 6 time points once the fish were removed. And for the state, what we did is that we filtered the water on 3 different filter sizes in order to understand which kind of particle sizes we could recover. We performed an experimental design last summer, so we had 6 temperatures ranging from 5 to 30 degrees. Inside each chamber we had 3 pH, so 1 alkaline, 1 neutral and 1 acidic. And inside each tank we had 4 fish that were inside 10 liters of water. So that was a kind of lot of work for sampling, so we generated 1,100 samples, and currently we are working on the eDNA and RNA QPC analysis using primers and probes that are specific to the goldfish. So here I just wanted to show you a small glance of the kind of analysis we want to perform. So here you can see the levels of eDNA and RNA molecules with time. So in hours during the degradation time, so once the fish were removed. On the upper panel, this is for eDNA. On the bottom panel, this is for RNA. And what we are interested in is, for example, to understand what are the differences in decay rates between eDNA and RNA, between temperatures that you can see here on the left at 10 and at 25 on the right, and also differences between pH that you can see here in different colors. And what we want to do ultimately is to build predictive decay rate models depending on the kind of molecules. So that's all for me. I just wanted to tell that if you have any questions, if you want to talk about this project with me, I would be very happy to do that after this session. Thank you very much for your attention.